Dear students, in the earlier classes on connective tissue, you have learned what is connective tissue and what are the three constituents of the connective tissue, that is cells, fibers and the ground substance. We have also learned about the different types of cells of connective tissue, the three different fibers of connective tissue and the various constituents of ground substance. We have learnt about the classification of connective tissue into loose connective tissue, dense connective tissue, embryonic connective tissue, the sclerous connective tissue and the fluid connective tissue. In this lecture, you will be learning about the sclerous connective tissue. So the sclerous connective tissue is also known as the skeletal connective tissue, and why it is called sclerous is because of its hardness. There are two types of sclerous connective tissue. One is cartilage and the other is the bone. In this class, you will be learning about the cartilage. Before we start with the discussion of the cartilage, I am presenting a case scenario which is concerned with the topic that we are going to discuss. A 70 years male comes with a complaint of swollen and painful joints of hand as shown in this picture, and you can see the terminal phalanges which are bent and swollen. After seeing this case, you should be able to answer what is the probable cause for this condition and what structures are affected. You should be able to give the explanation for this condition. After listening to the lecture, you will be able to answer these questions. The learning objectives for this session are: at the end of the lecture, the student should be able to define cartilage, which we have already defined it as the sclerous connective tissue, the composition of cartilage, types of cartilage and description of each type with their location and examples, functions of the cartilage, growth of cartilage and the clinical importance of the cartilage. Coming to the classification of the cartilage, let us revise what you have learned in the previous classes. So the connective tissue can be classified into connective tissue proper, specialized connective tissue, embryonic connective tissue or mucoid connective tissue, the supportive or skeletal connective tissue, which we call it as the sclerous connective tissue, then the fluid connective tissue. Fluid connective tissue you will learn in your physiology classes. Coming to the connective tissue proper, it is divided into loose connective tissue or areolar tissue, dense connective tissue which is of two types, regular and irregular. Specialized connective tissue consists of elastic tissue, reticular tissue and adipose tissue. Under the embryonic connective tissue, we have learned about the umbilical cord, which contains the mucoid connective tissue and we call it as Wharton's jelly. Under the supportive connective tissue, there are two types. One is the cartilage and the other is the bone. Cartilage is a specialized connective tissue and it is the chief connective tissue of the body. It is firm but softer and flexible than the bone, and it is located in areas that require rigidity and elasticity. If you see the composition of the cartilage, it consists of cells, the extracellular fibers, the intercellular matrix or substance, which we call it as the ground substance. If you see the composition of the cartilage, it consists of cells. The cells of the cartilage are the chondrocytes, which you can see them in this picture. Then the fibers. The fibers can be collagen fibers or elastic fibers. The other constituent is the ground substance. It can be proteoglycans or glycosaminoglycans, and the ground substance is amorphous and it is gel-like. It also contains water and electrolytes. In this picture, you can see the constituents of the cartilage, that is cells and extracellular matrix. The cells can be chondroblasts, the young cells, and the chondrocytes, the adult cells, which can be seen in groups, and they are located in what is known as cell nests or lacunae. The extracellular matrix consists of fibers and ground substance. The fibers can be collagen fibers or elastic fibers. The ground substance is rich in proteoglycans and hyaluronic acid. Then the other feature which you are seeing here is the perichondrium. Peri means around, chondrium means cartilage. So around the cartilage. So the cartilage is surrounded by perichondrium, which is highly vascular. You are seeing so many capillaries in the perichondrium, and there are two layers of perichondrium, and they are the fibrous layer. The outer layer is called the fibrous perichondrium, and the inner layer is called the chondrogenic layer, which contains the chondroblasts. And the perichondrium is highly vascular, whereas the cartilage is avascular. The cartilage receives its nutrition by diffusion from the blood vessels or the vascular perichondrium. And then if you see the arrangement of the cells, you can see in them in the lacunae in this picture. Coming to the features of the cartilage, and it is firm and flexible. It has got good tensile strength, hence it bears the weight without bending, and this is due to the presence of extracellular matrix. The cartilage is supportive, and it is in the regions that require both rigidity and extreme flexibility. In those regions it is seen, for example, in the joints and pinna of the ear. And regarding the vascularity, the cartilage is avascular, and it receives its nutrition through diffusion from the perichondrium, as I have shown you in the previous slide. Because it is avascular, the regeneration of cartilage after the injury is poor, and because of the injury, a connective tissue scar will be formed. The innervation of the cartilage, it has no nerve supply, hence it is insensitive to pain, and it has no lymphatics. So the cartilage has no blood supply, no nerve supply, and no lymphatic supply. The chondrocytes are derived from the mesenchyme, and their function is they produce matrix and fibers, and they are placed in what is known as lacunae or cell nests. Here in this picture you can see them. They are in groups, isogenous groups, and they will be lying in cell nests or lacunae. And there are two types of cells you are seeing here. One is the small cells, the other is the large cells. These are the small cells you are seeing, and you can see the large cells. These small cells are metabolically active and they are capable of multiplication. And they are rich in mitochondria, endoplasmic reticulum and Golgi apparatus. They have got the euchromatic nucleus. So see the nucleus of the small cells here. This nucleus is euchromatic. And when you compare them with the large cells, these large cells are less active and they are not capable of multiplication. And the prominence of organelles is also less in the large cells when compared to small cells. The large cells secrete alkaline phosphatase, resulting in calcification of the cartilage, which is seen in the old age. The calcification of cartilage is seen in the old age, and the nucleus of the larger cells is heterochromatic. So compare the heterochromatic nucleus of the large cells with the euchromatic nucleus of the smaller cells. Coming to the matrix or intercellular substance, which gives firmness to the cartilage, and the composition of matrix is it contains 75% of water and then 15% is collagen fibrils, because of which there is tensile strength for the cartilage, and 10% proteoglycans will be present. They add resilience to the cartilage. And in this picture you can see the perichondrium and then the cartilage cells in the lacunae, that is chondrocytes in lacunae you are seeing. There are two types of matrix, that is territorial matrix and interterritorial matrix. The territorial matrix surrounds the groups of cartilage cells. You can see in this picture the cartilage cells in groups, that is called the cell nests, isogenous groups. These are the various terms that are used for describing the groups of cartilaginous cells. And the territorial matrix is a newly formed one and it is non-fibrous. And the interterritorial matrix, it is present between the cartilage cell groups. This is the interterritorial matrix, and the interterritorial matrix is Walder matrix and it contains the fibers, that is collagen or elastic fibers. So now you know that there are two types of matrix, the territorial matrix which is newly formed and non-fibrous and surrounds the cartilage cell groups. The interterritorial matrix, that is present between the cartilage cell groups, it is Walder matrix and contains the fibers. Coming to the ground substance, it is gel-like in consistency and it is basophilic in staining and it exhibits the property of metachromasia with PAS stains. It is positive. The composition of ground substance, it is rich in chondroitin sulfate, the keraton sulfate, the hyaluronic acid, electrolytes and water. These are the constituents of the ground substance. The hyaluronic acid is rich in weight-bearing areas of the cartilage, that is in the synovial joints, as it provides free movements in these joints. The cartilage provides support to the soft tissues, as you have seen in the trachea and larynx. It provides support to these two structures and it provides a sliding surface for the joints, as in the articular cartilage at the ends of the bones that come in articulation. For example, the knee joint, the lower end of femur and the upper end of tibia, or articular cartilages that provide the sliding surface for the movements of the knee joint. Then it forms a model for growth of a long bone. All the developing bones are cartilaginous and later on they will be replaced with the bone. Coming to the perichondrium. So all around the cartilage there is the perichondrium, and the perichondrium is a dense fibrovascular and irregular connective tissue that surrounds the cartilage, and you have learned that it is rich in blood vessels and it provides nutrition to the cartilage cells. So by diffusion the cartilage cells receive nutrition from the perichondrium, and the perichondrium is required for the growth of the cartilage. So if the perichondrium is removed, growth of cartilage will not take place. And in this picture you can see the perichondrium and you can see the chondrocytes in the lacunae you are seeing, and you can see the matrix. In this picture you can see the perichondrium with its two layers, that is the fibrous perichondrium and the chondrogenic layer of the perichondrium. So the outer fibrous perichondrium consists of the collagen fibers, the fibroblasts and undifferentiated mesenchymal cells that can get converted into chondroblasts, the blood vessels and nerves. The fibroblasts can differentiate into the chondroblasts or the cartilage forming cells. The inner layer of chondrogenic perichondrium, it consists of flat chondroblasts which can differentiate into the spherical chondrocytes, and they will migrate to the center of the cartilaginous plate. That's what you are seeing in this picture. And here you can see the same picture. You can see where what is cartilaginous matrix, you can recollect, and what is meant by isogenous groups of chondrocytes also you can learn, and the two layers of the perichondrium you are seeing. The perichondrium is absent in articular cartilages in relation with the joints, and in the costal cartilages in relation with the rib, and in the epiphyseal cartilage in relation with the developing bone. You have to recollect what is diaphysis, metaphysis, epiphysis and the epiphyseal rate of growth cartilage in the developing bone. And it is also absent in the fibrocartilage. So that is the one which you will find in relation with the intervertebral discs. You will find what is known as the intervertebral discs in between the two vertebral bodies, which contains the fibrocartilage. So in the fibrocartilage, the perichondrium is absent. Now you know the various places where the perichondrium is absent. And in the adults, the perichondrium contains only fibrous layer. There is no chondrogenic layer of perichondrium in the adults. Regarding the growth of the cartilage grows by two mechanisms. One is the appositional growth, the other is the interstitial growth. One is addition from the surface and the other is addition within the existing. So the appositional growth is by multiplication and differentiation of the chondrogenic cells into chondroblasts in the perichondrium, that is addition at the surface. The interstitial growth, the growth is by multiplication or mitotic division of the chondrocytes that will form the isogenous groups. Now you know what is isogenous groups. Try to correlate with that isogenous groups. So multiplication of chondrocytes. So they will form another group of isogenous cell nests. So it is within the existing by division and differentiation. So these are the two mechanisms of growth of cartilage. Look at the picture to understand about the appositional and interstitial growth. So appositional growth is growth from outside by cells of perichondrium. So additional layers of chondrocytes are laid down by the perichondrium and that will secrete the new matrix, and this causes increase in the width of cartilage model. The interstitial growth or the internal growth or inside growth by chondrocytes that will increase in number by mitosis from the center of the cartilaginous model. This causes increase in the length of it. So look at this picture. So the appositional growth, there is undifferentiated mesenchymal cells that can get differentiated into chondroblasts. As you see from the perichondrium, the chondroblasts. So the undifferentiated mesenchymal cells, they become the chondroblasts. And these will secrete the extracellular matrix to form the lacunae, and they become the chondrocytes. The chondroblasts will become the chondrocytes. They go on dividing to form the isogenous groups, and they will be spreading a part of the chondrocytes. So that is appositional growth, that is addition from the surface. Then coming to the interstitial growth. So the interstitial growth, the chondrocytes that are derived from the chondroblasts of the perichondrium, they will be going on multiplying and going on forming the isogenous groups. So each chondrocyte in the lacunae will lay down the matrix to form its own lacunae and spread apart. That's how they will be forming from within by the division and differentiation of the chondrocytes. Coming to the classification of cartilage. Based on the histological appearance and the components of extracellular matrix, the cartilage can be classified into three types. They are the hyaline cartilage, the elastic cartilage, and white fibrocartilage or just fibrocartilage. So their name itself is implying their structural composition. In the case of elastic cartilage, the elastic fibers are there. In the case of fibrocartilage, the collagen fibers are there. In the case of hyaline cartilage, it has got its name from its appearance. Hyalos means glossy appearance. So because of the transparent appearance, it is called the hyaline cartilage. Coming to the hyaline cartilage, it is the most common type of cartilage, and the word hyalos is derived from Greek, which means glossy. With hematoxylin and eosin staining, the matrix is basophilic. You can observe the color of the matrix. The perichondrium is absent in the hyaline cartilages in relation with the articular cartilage, the nasal cartilage, and in the costal cartilages. Epiphyseal plate of cartilage also does not contain the perichondrium. In this picture, you are seeing the hyaline cartilage. Observe the perichondrium, which consists of the fibrous layer. This is the fibrous layer. This is the chondrogenic layer, which consists of chondroblasts, which are elongated, you are seeing, and which gets transformed into the spherical chondrocytes when they reach the center of the cartilaginous plate. And here you are seeing the chondrocytes in isogenous groups or cellness with the territorial matrix and interterritorial matrix. In this picture, you can see the arrangement of a rim of perichondrium you are seeing here, with the fibrous and chondrogenic layers, and you can see the cartilage cells in lacunae in the isogenous groups of 2 to 4 cells, and these chondrocytes, they synthesize and secrete the type 2 collagen fibers. If you observe the matrix, it consists of homogeneous, highly basophilic, transparent and amorphous. These are all the features of the matrix. So which is homogeneous, highly basophilic, transparent and amorphous. The ground substance gives the glossy appearance, and it contains type 2 collagen fibers, not collagen fibers. So because the refractive index of these collagen fibers and the ground substance, which is rich in hyaluronic acid, the refractive index of these two are the same. That's why you cannot observe these collagen fibers in the matrix because of the same refractive index of the ground substance, which is rich in hyaluronic acid, and the collagen fibrils. So the hyaline cartilage contains collagen fibrils, not collagen fibers. This point you have to remember. This is a picture which you can see how to draw the hyaline cartilage, showing the 2 layers of perichondrium and the chondrocytes in lacunae or cellness or isogenous groups. These terms you have to repeatedly understand. Then the interstitial matrix you are seeing, and the same in the slide also. You can compare with the slide. The isogenous groups of cellness or lacunae and the matrix and the glossy appearance of it. So this is at a higher magnification where you can see the territorial matrix, which is basophilic and rich in sulfated glycosaminoglycans, that is chondroitin sulfate. There is chondroitin 4 sulfate, 6 sulfate are there. Then the dermaton sulfate and keraton sulfate. Then the interterritorial matrix, which is less basophilic, and it is glossy in appearance because it is rich in hyaluronic acid, and this contains the collagen fibrils. This is the collagen fibrils will be present because the refractive index of the hyaluronic acid and the fibrils are same. It gives the glossy appearance, and you have learnt that the territorial matrix is non-fibrous, whereas the interterritorial matrix is fibrous. This point also you have to recollect at this point of time. Yeah. Coming to the examples for the hyaline cartilage. So during fetal period, where do we find hyaline cartilage? During the birth to adolescent period, where it is present, and at all ages where it is present, we will learn. During the fetal period, the fetal skeleton contains the hyaline cartilage. So this is a fetal skeleton. You can see the head of the fetus. You can see the developing eyeball, and this is the head and neck region, and you can see the epiglottis, and this is the upper limb. Then you are seeing the vertebral column with the bodies of the vertebrae, and then the ribs in the thoracic region. This is the dorsal root ganglia which you are seeing. So the fetal skeleton is hyaline cartilage model, which later on gets replaced with bone. And from birth to adolescence, we will see the hyaline cartilage at the epiphyseal plates, which will be replaced with bone, and once the epiphyseal plate of hyaline cartilage is replaced with the bone, the growth and length of an individual will stop. at all ages, you will find the hyaline cartilage at the ends of articulating bones. So, as articular cartilage, you will be seeing the hyaline cartilage, but you have to recollect this articular cartilage is without perichondrium. Then in the thoracic cage, you will find the costal cartilages, which will be joining the sternum at one end, and at the other end they are attached to the rib. In the respiratory system, you will find hyaline cartilage in the nasal cartilages, the trachea and major bronchi. These are also made up of hyaline cartilage. The cartilage of the larynx. The thyroid cartilage is the best example for hyaline cartilage, and the other cartilages of larynx that are having hyaline cartilage are the cricoid cartilages. Then what about the growth of the hyaline cartilage? So it is both by appositional and interstitial growth. Coming to the functions of hyaline cartilage in different areas, at the articular cartilages, the function of which is to provide a smooth surface for the movement, and at the costal cartilages in relation with the ribs, the flexibility, the property of flexibility is provided by the hyaline cartilage, and in the respiratory system, it gives firmness to the tubular structures of the respiratory tract so that the lumen will be kept patent for the free movement of the air. The clinical application of the cartilage. So, because of the arrangement of cells in the form of cellness, there is low antigenicity of the matrix in the hyaline cartilage. The homogeneous cartilage transplantations are possible without rejection in the case of hyaline cartilage. Then coming to this picture, so which was not labeled, and you can see the two layers of perichondrium, then the cartilage cells arranged in groups with territorial matrix and interterritorial matrix, giving the glassy appearance for the hyaline cartilage. Hope you have understood about the hyaline cartilage. Now let us learn about the elastic cartilage. It is also called yellow elastic cartilage because it looks yellow in the fresh state. It is more flexible and elastic, and it has got predominance of elastic fibers, and perichondrium is present. In this picture, you can see the perichondrium with the two layers and the chondrocytes. When you compare with the hyaline cartilage, the cells are larger in size and they are not seen in isogenous groups of many cells, and then they are seen as singly or in groups of 2 or 3. The matrix contains the elastic fibers. See this slide here. So the chondrocytes, if you see here and compare with that of the hyaline cartilage, these are bigger in size and they are singly or in isogenous groups of 2 or 3. The matrix is rich in elastic fibers and few type 2 collagen fibers and little ground substance. And this cartilage will not ossify with age. It is the difference between the hyaline cartilage and elastic cartilage. The similarities are both are having the perichondrium, and if you see the arrangement of chondrocytes, they are singly or in small groups of 2 or 3 in the elastic cartilage, and they are more in the hyaline cartilage. And the matrix contains the elastic fibers and few type 2 collagen fibers in the elastic cartilage. So there is no ground glass appearance as you see in the hyaline cartilage, and it will not ossify with age. Then when we use a special stain, Orsin stain, the elastic fibers, they will be appearing dark black in color, and then they are thinner at the periphery, which you can see, because with the special stain only you can see the individual elastic fibers you can identify, and they are thin at the periphery and thicker and shows branching in the interior of the cartilage. So this is a special staining of Orsin for the elastic cartilage. And you know that the function of the elastic cartilage is to give shape and support with elasticity. This is another picture of elastic cartilage. This is from pinna of the ear, where you are seeing the hair follicles in this picture. And this is the perichondrium with 2 layers, and then the central cartilaginous plate containing the singly placed large chondrocytes with the elastic fibers, which are seen in between the groups of cartilage cells or individual cartilage cells surrounding them, and the ground substance is less in this cartilage. And the growth of the cartilage is by both interstitial and appositional growth. Coming to the location of elastic cartilage, the examples for elastic cartilage are the pinna of the external ear, the external acoustic meatus, the auditory tube, and in the larynx, the epiglottis. The corniculate and cuneiform cartilages are the examples for elastic cartilage. In this picture, we can see the elastic cartilage with the 2 layers of perichondrium and the larger cells of the elastic cartilage, either singly placed or in groups of 2, and between these groups of cells or surrounding the individual cells, we will be seeing the fibrous matrix, which consists of elastic fibers with little ground substance. Coming to the white fibrocartilage. It is called white fibrocartilage because it looks white in the fresh state, and it consists of collagen fibers which are arranged along the lines of stress. So you will find the collagen fibers running in different directions as they are along the lines of stress of this cartilage. The collagen fibers are of type 1 and type 2 in this cartilage. The ground substance, if you observe, is inconspicuous except in the immediate vicinity of the cells. So you can see the chondrocytes which are singly placed, you are seeing, and you are seeing the collagen fibers. See the picture, you are seeing bundles of collagen fibers, and in between them the cartilage cells arranged in a row. That is what you are seeing, and there is an absence of perichondrium. That is one feature. You have not seen the perichondrium in this picture. So white fibrocartilage does not contain perichondrium. So the white fibrocartilage is a mixture of dense regular connective tissue and cartilage. The encapsulated cartilage cells, they are lying singly or in pairs between the bundles of type 1 collagen fibers. Here also you can see the drawing of the white fibrocartilage with bundles of collagen fibers, and in between row of chondrocytes you are seeing with little matrix. And what is the location in which the white fibrocartilage is found? The symphysis pubis is one region where you will find white fibrocartilage. In this picture, you are seeing the intervertebral disc between the bodies of the vertebra. This consists of the peripheral fibers known as the annulus fibrosus, which contains the collagen fibers. The central one which you are seeing is the nucleus pulposus. The other example for the white fibrocartilage is at the manubriosternal joint, that is the joint between the manubrium and body of the sternum. The other locations where you can see the white fibrocartilage are the articular disc of the temporomandibular joint and in the labrum glenoideus in relation with the shoulder joint, the labrum acetabulae in relation with the hip joint, and in the menisci of knee joint. So the menisci are white fibrocartilaginous discs. Then at the point of attachment of tendon to a bone is also white fibrocartilage. Again see the picture where you can observe the collagen fibers, and in between them you can see the chondrocytes either singly or in groups of 2 you are seeing. Then how to draw that white fibrocartilage? You can see how to draw the bundles of collagen fibers and in between them the rows of chondrocytes. In both these pictures you are seeing, and there is absence of perichondrium which you have to remember. So there is absence of perichondrium in the white fibrocartilage, but it is present in both hyaline cartilage and elastic cartilage. The white fibrocartilage offers resistance to compression and shearing forces, and it acts as a shock absorber. Then coming to the functions of the cartilage. The cartilage provides a model for the formation of new bone. And I was telling you, the developing bone to begin with is a cartilaginous model, and during development and growth it is converted into bone, and it provides support or flexible support to the soft tissues, especially in the respiratory tract, keeping the trachea open, and you will find it giving support in the external ear and in the nose. It provides sliding surface for joints, that is at the articulating ends of long bones, where it provides padding. Then it forms a structural basis for the growth of long bone as epiphyseal plate of cartilage, and it has got the shock absorber function in the intervertebral disc. Now let us see the clinical importance of the cartilage. There is what is known as chondrodystrophies. So that is disturbances in the growth and subsequent ossification of cartilage. The best example for it is the achondroplasia. Then the osteoarthritis, that is degenerative changes in the joints due to loss of articular cartilage because of its progressive degeneration and destruction, that is known as osteoarthritis. Then the traumatic rupture or detachment of cartilage due to injury. The more common is in the knee menisci that are affected by injury, and they can go into rupture or detachment. It is treated by partial replacement. There is what is known as costochondritis. Costo means rib, chondro means cartilage. So it is inflammation of cartilage in the ribs. This is the common cause for chest pain. Then herniation of intervertebral disc, and you know the intervertebral disc contains the white fibrocartilage and it is seen as the annulus fibrosus because of the compression of the intervertebral discs. It can result in herniation of its contents. When they get herniated, they show compression on the adjacent nerves which causes back pain. The other importance of the cartilage is the tumors of cartilage. It can be benign, which are known as chondromas, and if they are malignant, they are known as chondrosarcomas. So, see the picture of an achondroplasiac fetus. So, achondroplasia is an autosomal dominant condition. There is defective formation of protein in the cartilage. There is reduction in the proliferation of the cartilage cells in the epiphyseal plates of long bones. So because of that, there is shortening of the limb and short stature. So now you understand the importance of that epiphyseal plate of cartilage, which is a hyaline cartilage. Osteoarthritis. It is a disease of old age and it is a disease of articular cartilage. The articular cartilage of interphalangeal joints of hand, hip and knee becomes thinner. There is reduction in the intercellular matrix due to altered chondrocyte metabolism. The glycosaminoglycan content of ground substance is reduced and the water content is increased. There is loss of articular cartilage leading to the contact of articulating bones in a synovial joint with deterioration in movements of the joint. So, for example, in the knee joint, there is loss of articular cartilage with age that results in contact between the articulating bones that reduces the movement in the joint. Then see the picture and see the swellings at the proximal interphalangeal joints and distal interphalangeal joints. In the proximal interphalangeal joints, there will be hard bony outgrowths or gelatinous cysts will be seen, and they are known as the Boucher's nodes. And in the case of interphalangeal joints, if there are the same hard bony outgrowths or gelatinous cysts, it is known as the Heberden's nodes. So Heberden's nodes are seen at the distal interphalangeal joints and the Boucher's nodes at the proximal interphalangeal joints due to osteoarthritic changes in the articular cartilage. So the cause for this is formation of the calcified spurs in the articular cartilage. And in the beginning of the lecture, I have presented a case scenario of a 70 years male with a complaint of swollen and painful joints of hands. You can observe the distal interphalangeal joints that are swollen, and we have put some questions. That is what is the probable cause, what structures are affected, and what is the explanation for this condition. After studying about the cartilage, now you will be in a position to answer these questions. So, the probable cause is osteoarthritis. The structures that are affected are it is disease of the articular cartilage of distal interphalangeal joints. That's why this is called Heberden's nodes. Then the explanation is there is reduction in the intercellular matrix and glycosaminoglycan content, and there is increase in the water content. Loss of articular cartilage leading to contact of the articulating bones. Then it will further result in deterioration in the movements of the joints and the appearance of bony spurs or gelatinous cysts at these joints. Now you got the explanation for the clinical case. See, you can see the further progression on this osteoarthritis. Then in the case of knee joint, which we have mentioned as an example, see the exposed bones because of loss of the articular cartilage and the formation of bony spurs in osteoarthritis of knee. In this picture, you are seeing the rupture of the menisci in relation with knee joint, which we call it as the meniscal tears. So the traumatic rupture or detachment of the cartilage is the other clinical importance of the cartilage, which is more common in the knee, and the treatment is by partial replacement. The herniation of intervertebral disc. So these are the bodies of the vertebra which you are seeing, and in between the bodies of the vertebra you are seeing the intervertebral disc, which consists of peripheral annulus fibrosus, which is a white fibrocartilage, and central nucleus pulposus. So because of the compression of the intervertebral disc, the contents of the intervertebral disc, they herniate and they can produce pressure effects on the adjacent nerves, which is responsible for the back pain. In this picture, you can see the herniated intervertebral disc putting pressure on the spinal cord. Then one question that comes to our mind: why tissue rejection does not occur with cartilage transplantation? The homogeneous cartilage transplantations can be done without tissue rejection because the cartilage cells are located in cell nests. There is lower antigenicity of the cartilage matrix, and the matrix acts as an immunological barrier and prevents the entry of lymphocytes or immunoglobulins. That's why there is no tissue rejection with cartilage transplantation. Now, after learning about the cartilage, the different types of cartilage, the constituents and the structure of different types of cartilage, let us see what are all the points that will be of help in identifying these three cartilages in the histology lab. So this one is a picture of the hyaline cartilage, and this is of elastic cartilage, and this is of white fibrocartilage, and you can see the perichondrium is absent in the white fibrocartilage. The other two cartilages are having two layers of the perichondrium. And regarding the arrangement of the cartilage cells, they are seen in isogynous groups of 2 to 4 cells or more cells with territorial matrix and interterritorial matrix in the case of hyaline cartilage, and the glossy appearance of hyaline cartilage. In the case of elastic cartilage, the cells are larger in size and they are singly or in groups of 2 they are arranged, and around them you are seeing the elastic fibers. In the case of white fibrocartilage, the collagen fiber bundles are seen along the lines of stress, and in between them will be a row of cartilage cells either singly or in groups of 2. So, how to draw the 3 types of cartilages you know. Now, what are the other differences which you have learnt here? With age, it undergoes calcification and ossification, that is the hyaline cartilage, whereas elastic cartilage and white fibrocartilage, they do not undergo calcification and ossification with age. And what are the examples for hyaline cartilage, which is the most common cartilage? It is in the respiratory passages, the costal cartilages, the nasal cartilages, the articular cartilages, and the developing bone. Whereas for the elastic cartilage, all E's it will start, that is the external ear, the external auditory canal, the eustachian tubes, and epiglottis. The examples for the white fibrocartilage are the secondary cartilaginous joints, that is the symphysis pubis, the manubriosternal joint, and the intervertebral disc, that is the midline joints. Then the other examples for white fibrocartilage are the menisci of knee joints, the articular disc of temporomandibular joint, the glenoidal labrum, and acetabular labrum. Hope you have understood in detail about the various types of cartilage.